In lab, we're going to be using the Phenol Red Carbohydrate Broth tube test. Follow your directions. Phenol Red comes commercially prepared, and in the Phenol Red broth, you can add different carbohydrates. We're going to be using this test to differentiate between members of the family Enterobacteriaceae. So there's 3 carbohydrates that we're going to use to distinguish between the microorganisms we're working with. We are going to be using the Phenol Red glucose tube. And remember, our tubes, the caps are often very loose. So remember, when working with your tubes, to always pick it up by the tube and not just by the cap so it doesn't fall. So glucose, we're going to be using sucrose, and then Phenol Red lactose, and they're already labeled for you. You're going to label them on the glass with your initials, the date, and your microorganism. If you notice in these tubes, they have a small inverted test tube, which is called a Durham tube. We're going to be using the Durham tube to see if our microorganisms, when they go through fermentation, if they create gas. So the gas is going to be in the Durham tube. So when you're working with these tubes, you don't want to jiggle them around. And so you want to move very smoothly because you don't want to artificially introduce a gas bubble into the Durham tube. So each person or each mini group is going to get one of each of the carbohydrates, and then you're going to receive one plate that has a microorganism. The microorganisms we're testing today are Escherichia coli, Pseudomonas aeruginosa, Proteus houseri, and Enterococcus faecalis. So we're going to be transferring aseptically, going from a plate to the Phenol Red carbohydrate broth tubes. So once you've labeled your tubes, remember, go ahead and take your inoculating loop, and we're going to flame it from the base to the loop. And remember, you're aiming for the top of that inner blue cone, the hottest part of the flame, and you want to leave it there until your entire loop glows orange, and then pause for a moment. Remember, microbiology is fun. You're going to invert your plate, and we're going to use the lid as a shield. And go ahead and select a colony. Close your plate. Grab your Phenol Red carbohydrate broth tube, and remember, you use your pinky. You're going to remove the cap. You hold it horizontally. You're going to flame the lip. 1, 2, 3. And then you're going to go in, move the tube, make sure your loop goes in the broth, and then you just want to give it a little shake to dislodge some of the bacteria off of your loop without touching the sides. Remove the tube. Remember, you flame again. Recap. You have to flame your loop to sterilize it. So from the base to the loop, leave it there till it glows orange. Microbiology is fun. And then you can place that on the lab bench. Now, to disperse the bacteria into the tube, you're just going to take your Phenol Red tube and you're just going to gently roll it between your palms, and then it's ready to incubate. We're going to be incubating it at 37 degrees and for 18 hours, so just overnight, and then we will read the results in lab next period. So when you're done inoculating all 3 of your carbohydrates, so glucose, sucrose, and lactose, then take your tubes and place them in the rack at the front of the classroom.